Good morning. Today we are going to be talking about vision. Vision is super cool, super complicated, but I will do my best to explain it. So I think I fixed the audio problems so that it should be smoother audio. I changed the, I changed the settings to my headset microphone, and I thought it automatically changed that, but it didn't. So I'm hoping you don't get the background noise or the sound of the pen writing on the screen. Are we frozen? That is the real question. Whoa. What the heck? Okay. I guess we're good. Oh, I don't like that. Show app icons. Okay, we're good. Cool. So something I want to talk about is this idea of Gestalt. I don't know if you heard of Gestalt psychology, or the theory that the whole that we perceive is something different than the parts, or the sum of the parts, or whatever. And what's really interesting about vision is we're hallucinating pretty much every day. So it makes you think what happens, you like drop some acid or something. And people call that hallucinating, but we're already hallucinating every day. I don't know. So this is a cool quote. A lot of these slides I got from Dr. Haley O'Brien, so I want to give her credit and thank her for letting me use her slides. This is a quote. It says, "Our actual experience of visual environment is a fully analyzed opinion about what the brain thinks is out there." So your brain is kind of just making up your surroundings. I forget where I heard this quote, but I really like it. It goes, "We create the world just by looking at it." And neurophysiologically, that's actually true. Pretty cool stuff. This is an example of Gestalt. So over here, there's a bunch of shapes and lines, and we have no idea what it is. But then if you rearrange those into something recognizable, our brain fills in the gaps. This is really interesting. So these are kind of all your retinal spots that we pick up, and all these spaces in between here we literally can't see. They don't exist, which is wild. All these spaces, a huge portion of our vision doesn't exist. And the only reason it exists is because your brain just uses context clues to just fill in the gaps. And that happens a lot in the visual association areas, which I'll show a picture of. But it's really cool stuff. This was too good not to use. This is from Dr. O'Brien's slides again. It's a good... Props to you if you get the joke. When you see this in her lecture in neuro, just laugh with everyone else like it's the first time you're seeing it, so I don't get in trouble for using it. But no, she probably won't care. So when anything you see, when it travels to your retina, like the back of your eye, the image is inverted vertically, right? And reversed horizontally. So this corner ends up here. This corner ends up here, this corner ends up here, and this corner ends up here. This was really hard for me to visualize. It still is. So I'm going to keep it on the bottom right-hand corner in these next couple slides. The visual pathway, don't get too hung up on the details, but I do want you to know the order, where it travels, and the order of where it travels, how it gets from our visual field to the visual cortex. And you'll also see this called V1. V1's like primary visual cortex, and then the association areas are like V2, V3, V4, etc. That's different from cranial nerve 5 ophthalmic branch, which is also called V1, but I like this next picture better, so I'm going to use this. Okay, so each eye has its own visual field. So the right eye has what's called, you could call it the right visual field or the temporal visual field, because they split up this terminology into nasal and temporal. So just think where your nose is, and then think where your temporal lobe is, which is like the lateralmost lobe of your brain. Then in this, in red, would be the nasal field of the left eye, or the right field of the left eye. Does that make sense? And then this would be the nasal visual field of the right eye, and this would be the temporal visual field of the left eye. So remember, Miss Yellate here, everything is flipped and reversed. So if ever in doubt, it's just the opposite. So the left, or the temporal visual field of the left eye, crosses over and hits the nasal half of this retina, and vice versa. The way I like to think about it is, you have in red, you have your two right-sided visual fields, and to make sense of that information, because one right-sided visual field is coming from the left eye, one right-sided visual field is coming from the right eye. To make sense of that information, those visual fields have to reunite at some point before getting back to the cortex. So, in order for them to reunite, in red right here, for instance, the temporal visual field of the right eye goes straight through the optic chiasm, and then the nasal visual field, or the right visual field of the left eye, when I said continue straight through, this one crosses over the optic chiasm, and then this one stays on the same side. So that's the way I like to think of it. These 2 points are good to remember. Like I said, the image on the retina is inverted, and then the cerebral cortex back here receives the encoded image of the contralateral visual fields of both eyes. So this is the left side of the brain, right? And since we reunited the right-sided visual fields from both eye information, it's the contralateral visual field of both eyes that ends up going to the primary visual cortex. Okay. Anyway, let's continue with our neuronal tract. So I got hung up there. Anyway, so on the left optic tract, right after the optic chiasm, you have a continuation of the right visual fields from both eyes. Lateral geniculate body. You also see lateral geniculate nucleus. And I'm just going over one side to make it simpler, but remember, like, this blue side's happening on the other side, okay? And then after synapsing here, the second order neurons travel in something called the geniculo-calcarine tract. And geniculo comes from the name, geniculate body. Calcarine comes from something called the calcarine sulcus. And that is the primary visual cortex. It's a sulcus in the brain that demarcates where it's at. So takeaway here, the image is flipped in reverse from the retina. At first, the message is only unilateral, and then you have to reunite the right side in red at the optic tract, or the left side visual field of each eye in blue. That gets reunited at the optic tract, goes to the geniculate body, lateral geniculate body, then the geniculocalcarine tract. You'll also see these called optic radiations. The optic chiasm is like super high yield because the temporal fields, visual fields of both eyes cross at the optic chiasm. If you follow it, this blue crosses here, and then the red crosses here. They can ask like tons of questions on it. There's a lot of things that can compress the optic chiasm. The most common things are like pituitary tumors, craniopharyngiomas. This will most likely be in adults in the question stem. This will be kids. Aneurysms can compress the optic chiasm. A lot of stuff. And, oh, sorry, I didn't even get to the high yield point. If that's compressed, you get something called, and you may have heard of this, bitemporal hemianopsia. And all that means this is knocked out, this is knocked out. So the temporal fields, right, bitemporal, the temporal fields of both eyes, you can't see. So these people actually, a classic question stem will say these people are bumping into things because it messes with your peripheral vision. Trying to remember, that's like a big one. Vision problems, or even they'll straight up tell you that the patient has trouble seeing out of the lateral fields of both eyes, and that's just a dead giveaway that something's compressing the optic chiasm. They love optic chiasm questions. They love it. I think I love it too. This is, you don't have to know any of this, but this is just to show you the complexity of these association cortices. And the primary visual cortex, that's not how we actually see. The association areas are how we see and make sense of the world. V1's kind of just a catalog, right? So there's tons of V2, V3, blah, 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 like so many association areas. The extra striate cortex, some people say that's responsible for the relationship of like what we see and like consciousness and what makes us human and all that stuff. So yeah, vision's very, very complicated. Okay, so something crazy about color, and you guys may already know this, but we can only see 3 colors, which is wild. We only have 3 types of photopigments, iodopsin, for blue, green, and red at different wavelengths. And how we see everything else is probably just like some combination of these, or our brain like filling in the information again. I don't know all the details, but that always like blew my mind, that we can only see 3 colors. And this is where like the red-green color blindness comes from. You've probably heard of that. Cones, C for color. It's easy. There are far fewer 6 million cones than there are rods, 125 million. Rods respond to low light, so that's like black and white vision. The photopigment is actually like a purple blue, but when light hits it, it ends up looking like black and white to us. As you can see, like the color is kind of like this area. maybe like a purple, blue, green, I don't know. And they only have one type of pigment. It's called rhodopsin. There's 3 types of iodopsins in the cones. The fovea is an area that is highly, highly densely packed with cones and actually has no rods. So when you hear the term foveating, just think of like focusing. Like that's where the fovea is what allows us to focus on a single spot, have a lot of like 20/20 visual acuity. That's cones. Bipolar cells, I'll talk about that on the next slide, I think. Now for the good times. Okay, so I don't want to spend a lot of time on this because it sucks. Basically, there's, just giving an example of what happens, how phototransduction actually happens. Cyclic GMP is involved. There are sodium channels involved, calcium channels, potassium channels. Something interesting, this membrane potential. So what do you notice about that? It is far less negative than what we normally think of, which is like what, negative 70 millivolts for membrane potentials. And that is because there is a ton of densely packed sodium potassium pumps, among other things, but that's a big reason why. And there's more leak channels as well. There's like a lot more channels in this area. So anytime I get a chance to relate two areas, so remember this was like week 1 material and this is week 3 material. So anytime I get a chance to tie in 2 things together, that's always a good opportunity for a test question. Because it's the ideal situation for retention. So I will say that something weird about phototransduction is that it's depolarized in the dark. So it's kind of quote unquote turned on in the dark. And there's this tonic, which kind of just means like default release of glutamate. Our lab studies glutamate a lot. It is normally an excitatory neurotransmitter, but in this case it's kind of inhibitory. So let's look at what happens when light hits. When light hits, these intracellular signaling changes happen. There's the activated retinal. It changes, remember, the cis and transforms. The opsin gets bleached. Don't really know what that means. It's like bleached, actually. Transduction gets activated, and that's easy to remember because it's phototransduction. There's a really complicated intracellular cascade. Cyclic GMP was high over here. Cyclic GMP gets decreased. These sodium channels close. This is probably the most important part of what happens when light hits, because sodium channels close. A positive ion can't get into the cell, remember, so that causes membrane hyperpolarization. Because if a positive ion can't get in, you're going to get more negative on the inside relative to the outside, right? Because remember, membrane potential is intracellular voltage minus extracellular voltage. Also, potassium channels leave, so you have a positive ion leaving. That further contributes to the hyperpolarization. Now, when the hyperpolarization occurs, you have a decrease in glutamate release. And it's actually this inhibition, or the decrease of glutamate release, that allows for the full phototransduction to occur. And remember, the light's coming in out here. It kind of like skips everything, goes to the back of the retina, remember, because this is where the photoreceptors are in the back. So these are like rods and cones back here. These are bipolar cells. And these kind of remind me of the DRG because they have 2 poles. They have two arms, reaching out. One arm communicating over here, one arm communicating over here. And it's actually the bipolar cells that are really important for getting the message to the ganglion cells. Ganglion cells, which are in yellow up here. Okay, so this is, remember we talked about graded potentials. So you have these photoreceptor cells transducing, so they're transforming an environmental stimulus into a voltage change. That voltage change right here is still just a graded potential. So until we get to the action potential, all these membrane changes are doing is making it more likely that the action potential will occur. Okay, so the information, light comes in, all those cyclic GMP stuff happens, sodium channels close, glutamate release decreases, and you start to have a graded potential, information path, and then the bipolar cells are the first to know that they can relay the message in the form of a voltage change to the ganglion cells. And then up here we have the action potential happening, and the ganglion cells actually are the cell bodies of the optic nerve. Does that make sense? So then the message goes to the optic nerve and then to the brain. And then to relate it back to this, optic nerve right there. So then we have the beginning of this process. Hope I tied that together okay. Wikipedia is the best. I studied so many hours using Wikipedia. Yeah, they take their stuff like directly from textbooks, and it's very reliable. I even use it for like my PhD work because they have good references at the bottom. Then I click, oh my gosh, I hope you guys don't hear that and get bombed with it. Anyway, so this is just a summary of what happens with the weird situation. Just remember, it's the photo, or the whole situation kind of turned on in the dark, and then it's slowed down when light hits. The glutamate release is inhibited, hyperpolarization happens, and then phototransduction officially occurs. So why, right? Why? I can tell you, you will see a lot of questions about these two things. And they're pretty much the same thing. Remember, adenylyl cyclic AMP. They're the same thing, just the nucleotide's different, right? Guanylate instead of adenylate cyclase. The two highest yield pathways that are involved in is what we just talked about, visual stuff, and then this is probably like even more important, relaxation of vascular smooth muscle throughout the body. Remember when I talked about the mechanism of vasodilation at blood vessels? Endothelial cells, remember they have these muscarinic receptors. Oh, I hate when that happens. Muscarinic receptors, acetylcholine binds, parasympathetic nervous system. Basically, you end up getting nitric oxide. The mechanism by which nitric oxide relaxes smooth muscle and produces vasodilation is through intracellular signaling involving guanylate cyclase and cyclic GMP. And there are a ton of drugs that act on these processes. Well, I mean, for one, like nitrates for angina, like heart attacks and stuff, like nitroglycerin, right? That acts via this pathway. Viagra, sildenafil. What else? Anyway, there's tons of cardiovascular drugs that hit this pathway, so it's very, very likely you'll see a question like that on board. So that's why it's important. Pupillary light reflex. This is also very high yield. Let's ask a test question on this. So the afferent limb, be familiar with these terms. Afferent limb or pathway, you'll see that too, and the efferent limb. If you can keep this straight, you can get any question right. Okay, it's actually simpler than it seems. The first thing I'm going to do is go over the pathway. So we have light coming in. The afferent is sensory, right? Remember, and it's going back toward the brain, and that is the optic nerve, cranial nerve 2. Optic, and that makes sense, right? Optic nerve receives the light. That's kind of what we just talked about, right? Afferent pathway travels back through the optic nerve. Information crosses over to the contralateral pretectal nucleus, or pretectal area, and this is in the midbrain. Midbrain. Oh, gosh, it's annoying. I just want to close that off. Next, this is really important. From there is bilateral projection. Bilateral projection. Bilateral projection. Bilateral projection to the ipsilateral and contralateral Edinger-Westphal nucleus. If that sounds familiar, it's because it's the cell bodies for cranial nerve 3, which will end up being the efferent or motor limb. Oculomotor, right? It's easy to remember. Wow, struggling. From the Edinger-Westphal nucleus, and we're just doing one side right now. It's as if the light got shown in the left eye. So remember, contralateral, so right pretectal nucleus, then bilateral projection to both ipsilateral and contralateral Edinger-Westphal nucleus. From there, it's pretty straightforward because there's no more crossing over, right? There's synapse. The right Edinger-Westphal nucleus sends its axons in the form of cranial nerve 3 out to the right eye, and because of the bilateral projections from here to here, we also get oculomotor activity to the left eye, which was the one that received the light source. So that's the reason when you shine light in one eye, both pupils should constrict. Okay? Because remember, cranial nerve 3, we're talking parasympathetic. Kind of a stupid way that I remember this is Edinger-Westphal. You'll see EW, like ooh, and then you can pretend like there's a tiny, you kind of hold your hand out in front of you, pretend there's like a tiny like ant that you're squeezing, and then you close your fingers together, like kind of pupils constricting you. I don't know, whatever works for you. But this is also, what else do I want to say about this? Pretectal nucleus, Edinger-Westphal nucleus. Oh yes, like pathologies. Okay, so we know a lot of test questions will ask you which limb is affected when they give you patient results from an eye exam, right? So if you shine a light in a patient's eyes and nothing happens in either eye, there's no response at all, you know this message is never getting past here. So you know the afferent limb is affected. If you shine the light in the left eye and the left eye responds normally, but the right eye has no reaction at all, you know that the right side efferent limb is affected because you're receiving the light information and responding normally in the left eye, but this message can't get to the right eye to tell the pupil to constrict because that's the efferent limb is messed up. There's something called the Marcus gun pupil, and this is really common because you see this a lot in third nerve palsy. We call it third nerve palsy in diabetics. And anytime there's ischemia to the third nerve, which is the efferent limb, you get something called a relative afferent pupillary defect. And what that means is it's called a swinging flashlight test. And they'll shine the light in, let's say, the left eye and then swing it over to the right eye. And whichever relative afferent. Yeah, so when you shine it in the unaffected eye, wait a second, this always confused me. Don't worry about that for now. I'll explain it next lecture when I Wikipedia it. This is a good video on the pupillary light reflex. Okay, let's do a practice question. A 5-year-old girl is brought to her pediatrician because her mother says she's frequently bumping into stationary objects while playing. Visual field examination tells you straight off bilateral peripheral vision field defects. So right away you know the optic chiasm is compressed. CT, the head reveals calcifications in the pituitary fossa, which the following is the most likely origin of this child's brain tumor. I'm not expecting you guys to know this stuff. I just wanted you to recognize what it looks like with when they're presenting bitemporal hemianopsia. Rathke's pouch is like the embryological origins for craniopharyngioma, and it always has calcifications because the embryological origin is kind of like an up-pouching of the mouth. So I just think like, oh, teeth is normally there, calcifications, and that's why there's calcifications up there where they're not supposed to be. But you don't have to know that. I was just giving you guys an example. Alright, we will talk about CSF next. The lectures after this will be shorter. I wanted to get the dense stuff out of the way first. So sorry for all this material. I know it's a lot. But you will see this again. And neuro is really, really good to be exposed to this stuff before because a lot of students struggle in neuro. So good luck.